Hi, my name is Ravana. Let's explore the process of going from DNA to protein in more detail, along with examples. DNA transcription. 1. Example, consider a gene in human DNA that codes for the protein insulin. 1. The DNA sequence of the gene contains the instructions to make insulin. 2. During transcription, the DNA unwinds, and an enzyme called RNA polymerase binds to the DNA at the promoter region of the insulin gene. 3. The RNA polymerase synthesizes a complementary mRNA molecule by pairing the nucleotides with the DNA template strand. 4. For example, if the DNA strand has the sequence ATCG, the resulting mRNA sequence would be UAGC, since RNA uses uracil, U, instead of thymine, T. 4. The mRNA molecule is synthesized until the RNA polymerase reaches a termination signal on the DNA. mRNA processing. 1. Example, the newly synthesized mRNA molecule undergoes several modifications. 1. Let's continue with the insulin example. 2. In humans, the pre-mRNA undergoes splicing, where introns, non-coding regions, are removed, and exons, coding regions, are joined together to form the mature mRNA. 3. After splicing, a modified cap and a poly-A tail are added to the mRNA molecule to protect it and facilitate its transport out of the nucleus. Translation. 1. Example, the mature mRNA carrying the instructions for insulin synthesis now moves to the cytoplasm, where translation occurs. 2. Ribosomes, consisting of RNA and proteins, bind to the mRNA and initiate the process. 3. The ribosome reads the mRNA codons in sets of 3 nucleotides. 3. Let's assume an mRNA codon sequence of UAUCU triple AGGUCAG. 4. Transfer RNA, TRNA, molecules, each carrying a specific amino acid, bind to the ribosome and pair with the corresponding mRNA codon through their anticodons. 5. For example, a TRNA with the anticodon AAU pairs with the UA codon and carries the amino acid leucine. 5. The TRNA with the anticodon AGA pairs with the UCU codon and carries the amino acid serine, and so on. 6. The ribosome catalyzes the formation of peptide bonds between the amino acids brought by the TRNA molecules, creating a growing polypeptide chain. 7. This process continues until a stop codon is encountered, indicating the end of protein synthesis. Protein folding and modifications. 1. Example, once the polypeptide chain is synthesized, it undergoes folding and potential modifications to form the functional protein. 1. Let's consider the insulin protein. 2. Insulin undergoes a complex process of folding, involving the formation of disulfide bonds between certain amino acids. 2. The folded insulin protein adopts a specific three-dimensional structure, allowing it to function properly. 3. In addition to folding, proteins may undergo post-translational modifications. 3. For example, insulin undergoes proteolytic cleavage, where a portion of the protein is removed to produce the active form of insulin. Protein function. 1. Example, the fully folded and modified protein, such as insulin, performs its specific function within the cell or organism. 2. Insulin, for instance, plays a crucial role in regulating blood sugar levels by facilitating the uptake of glucose into cells. It's important to note that the examples provided here represent a simplified view of the complex processes involved in DNA to protein synthesis. The actual mechanisms can vary depending on the organism and specific proteins being synthesized.